Good evening, friends and colleagues. So lovely to see all of you here tonight. I would like to begin by acknowledging the traditional owners of the lands on which we meet today and pay my respects to elders past, present and emerging, and recognise their very significant and ongoing contribution to local and global society. As one of Australia's leading research-intensive universities, the University of Queensland is dedicated to research impact through excellence in both discovery and applied research. The exceptional quality of our research is evidenced through multiple international ranking systems, with UQ consistently in the top 1% globally for research impact across a large number of disciplines and fields. Of relevance to tonight's event include human biological sciences, clinical medicine and biotechnology. The outcomes of our research speak for themselves. This includes new knowledge captured in hundreds of thousands of research publications, over 18,000 PhD graduates, over 200 fellows of learned academies, over 360 granted US patents, over 100 startup companies associated with the commercialisation of products, including drugs, diagnostics and vaccines, along with important changes to health policies, guidelines and protocols. Together, these outcomes produce health, social and economic benefits, fulfilling our mission as a university to deliver for the public good. Indeed, UQ researchers have saved lives through groundbreaking discoveries and contributed significantly to the $3.32 in economic return for every $1 spent on research in Australia. In summary, we are very good at what we do. We deliver amazing outcomes for the local and global communities. Given all of this, I am enormously proud to be hosting this year's Douglas Ormonde Butler Memorial Lecture. The prestigious lectureship was established in 1977 thanks to the very generous bequest from the late Douglas Ormonde Butler. Its broad purpose is furthering genetics knowledge and understanding, particularly in relation to agriculture, livestock, and the human form and behaviour. The title of this year's lecture is Destiny versus Disease, where our experts will explore the role of genetics in disease risk, disease progression and disease outcomes. This year's event features a panel of distinguished UQ researchers, each with unique genetics expertise and insights tied to human health. They will showcase just some of UQ's research on genetics, disease susceptibility, human evolutionary response to pandemics, vaccines and personalised medicines.
What an incredible array of expertise we have up here, and I get to ask them all the questions. You also get to ask questions too. We've allowed a lot of time for that at the end, so make sure you think of them on the way through. Of course, tonight's discussion is about the role of genetics in, especially infectious diseases. I think we know intuitively that genetics plays a really important role in genetic diseases, but Loic, how much of a role do our genes play as opposed to the environment when it comes to disease?
Okay, I guess I'm sitting next to you, so I'll get to start. Well, this is a question. Well, the answer to that question varies from disease to disease, and we have ways to measure that using quantities like numbers, like we usually call heritability. So a disease is heritable, but how much is it heritable can vary between 10% and 60%. So 60% is typically what we can estimate for a disease like type 2 diabetes, which is, I guess, comorbidity that is associated with some, Kurt is going to talk about COVID-19, but some infectious diseases as well.
To answer your question about how much genes contribute to disease, well, this varies from one disease to another, and we have a way of measuring that with a parameter or number that we usually call heritability. And so how much a disease is heritable is given by that number. So type 2 diabetes, I was talking about this one, has an estimated heritability of about 60%. So what this means is that if we look in this room, how much differences in our susceptibility to that particular condition, in this context, type 2 diabetes, how much does we differ from each other in that susceptibility is caused by our genes. And that's the number, it's 60%. Right.
And so, Trent, one of the things that kind of plays into this is not just the genes that are there in the genome, but also which genes are on and which are off, the epigenetics.
Yeah, so I think I'm loud enough by the sounds. So that's exactly right. So I think, one of the things that we've learned over our journey, especially around what this genetics revolution has taught us, is that we've been able to decode the kind of what we were saying in our discussion before, the hard bits of the genome, i.e. the direct DNA sequence that exists within all of us. I think one of the things that's become really apparent is that that's not the whole story. So when we think about our genetics and what the makeup is, there's a whole range of modifications that actually occur to our DNA that intricately control the subsequent gene expression and all of the other traits that exist beyond that hard coding. So that soft coding that sits on top of our hard coding is something that people on this panel are trying to decode, and something that's looking to play a really important role in disease.
Can I add to that? I don't know, now I'm feeling the pressure that mine's not working. So one of the really amazing things about this field of epigenetics within the broader field of genomics is it's really how your environment can impact your genetic code. And so if you go back in the history of science, when Darwin was around and there was all these theories about evolution, there was the Lamarckian theory of evolution. And the Lamarckian theory of evolution was that giraffes would get a longer neck because they spent more time reaching for the top branches, and then that would pass on to their offspring. And that was kind of laughed at a little bit. I feel sorry for poor Lamarck. But it turns out that's exactly what epigenetics is. It's that all the environmental factors can actually determine which of your genes get switched on, and that pattern of switching on and off actually passes to your next generation. And that's really significant. I mean, we were talking about diabetes before and infectious disease, and we can see, for example, that we know that obesity is a risk factor for a lot of respiratory virus infections. We've seen that with COVID-19. But actually what happens is obesity can actually imprint on your genome and change which genes are turned off and on, such that even if you lose weight, you still have greater susceptibility to respiratory infections than somebody who was never obese. It's not a reason not to lose weight. That's not where this is going, but it's more that your environment can directly influence your genes.
And then there's more than just our genes inside our body as well, Gabrielle. There's all of the microbiome that's in there too, and that has an impact on our disease susceptibility.
That's exactly right. We're only just starting to really uncover the diversity of the different microbes that interface with the cells that we have in the body. And it's those components and the metabolites that they generate that actually influence epithelial cells and immune cells and create these epigenetic imprints. And this is sort of a whole new area of research in understanding how that works and how that affects the susceptibility of individuals to different types of diseases. So it's hugely exciting to be able to try and dissect how that works. And then how we can use the genetics not just of ourselves, but of the organisms we interact with, whether they're viruses or the bacteria, to select those, to modify those so that we can create therapeutics that might alter the course of your disease process.
Microphones, am I right? What would it mean to have the information about the genetic code of the beneficial bacteria in our body?
There's this thought, and there's a few case studies where interactions with different treatments can enhance the prognosis of outcome if you have cancers. Or potentially you could work out how to imprint your immune cells so that then you had greater resistance to some of these infections. And some of those are in the adaptive immune cells where we can change the genetic code, or we have T cell receptors that recognise the components of the bacteria or viruses. But others are in innate cells where we really have this genetic imprinting, or what we call trained immunity, that sets the scene for how this sort of memory-like approach can happen from cells that don't specifically recognise a particular sort of pathogen. So there's this multi-layered defence system that we have. And what's really intriguing is now the genomics and the genetics allows us the capability of starting to look at whether we can target identification of disease states well before we can see them by classical approaches, and whether we can generate therapeutics that might be able to target those states before we have different sorts of destructive diseases going on in our bodies. So then if we can get closer to personalised medicine and also to this sort of preventative healthcare, which would be a much better thing for people and a better economic landscape for us to be living in.
I think we take for granted how much we are able to do these days with genomics, but sequencing genomes happen within our lifetime. Trent, can you just remind us of what an enormous amount of change we've seen in the last few decades?
I mean, I think back, you know, I was very fortunate, I think, to spend time in the US as we were sort of celebrating the achievement of the Human Genome Project. And, you know, Loic, you'll be able to correct me on the number of years. I mean, the cost at that time, I think, was around $1 billion. What, 10 plus years or?
Yeah, well, about 20 years.
20 years that we spent doing that with what we thought was the best technology that we would ever have. And today, you know, you can take a sample and take it into, you know, pretty much, I don't know, many, many labs around Brisbane, and we would be able to decode your entire genome in a matter of hours at a cost of less than $100 or?
Well, it depends how much accuracy you want, but I think, yeah, the reasonable number will be $500.
Yeah.
But $100 is going to be, it's just right around the corner.
So from $1 billion to maybe less than $100.
Yeah, to an expensive dinner with friends, yeah.
But it's not even that, it's that you look at what students in
Yeah.
high school science are doing, and this is a basic high school science experiment now. You know,
Exactly.
like I remember doing sequencing when I was a PhD student thinking, wow, this is amazing, like we're living on the, and now these year 8s are doing it and, you know, writing it up in a report and done. So it's
They don't even appreciate it.
I know. It's come a long way.
They're like, oh, I've got all this homework, sequencing genomes. So look, how good is our understanding of human genomes across the globe?
Well, I guess at this point in time, most of our genetic studies are still pretty much skewed towards, I'll say, northern, well, European descent, let's put it this way, from...
White people. You can just say white people.
I don't know. Right? No, I think actually it's not just about being politically correct. I think there is a lot of finding the right language there, because I think the skin color doesn't contain all the information, right? And maybe this is something we're going to talk about a bit later, but I'm excited about the possibility of studying admixed populations. So this is the loaded term that we use in genetics to say, to designate people who have multiple ancestries. And in parts like South America or in South Africa, you can have someone who's white skin, but their ancestries are a lot more complex. And I think at the end of the day, this is what matters, because if, for example, we want to predict the risk of disease in someone who's admixed, which is one of the next frontiers, but even if we think about more homogeneous and underrepresented populations like, or here, indigenous Australians or Africa, other part of Africa. At this point in time, our ability to predict disease risk is essentially halved in those populations. So which means that today we can potentially make a life-saving decisions for someone with European ancestries and won't be able to do that for those populations.
So that genetic risk really does differ dramatically between races or between people of different ancestries.
So there are different layers to that. So the point to which the risk is different, which is something we can measure with epidemiological studies. We can see what's the prevalence of a particular condition in this group versus that group. But the other layer is how much those differences are due to genetic factors. And this is very hard to disentangle, because those groups will not only differ in their sort of genetic makeups, but they also have different environments or different cultural practices. And so essentially cracking that is one of the things I'm excited to do in the future.
How do you unpick that?
Well, I guess the place where I want to start is by studying admixed individuals. So those are people who have multiple ancestries. And the reason why I think this is a great avenue is because those people will have their multiple ancestries in their own genome, but those two or multiple genomes they have within themselves will always travel within the same environment. So if we can then understand why a particular gene or genetic variant they have inherited from their Irish grandfather, why this variant is acting differently or affecting gene expression differently as compared to the same variant or a similar variant inherited from their South African grandparents. But because we are immersed in the same environment all the time, we don't have to worry about cultural practices, etc. So this I believe is a great place to start, but those are very complex questions.
That's so fascinating. I think when we're talking about genetics, we often come to the place of personalised medicine, which we can talk about. But Gabrielle, I'd love you to sort of build on that by talking about not personalised medicine to an individual, but to a particular group with a particular health need.
Yeah, I think that's really important. And I think before COVID, most of the genome sequencing material that we had was from Western or Caucasian populations, and it became very obvious during COVID that while that comprises a large number of people in the world, we're missing great segments, which will be parts of other people's ancestry, of the Asian community, our own Indigenous community, and Indigenous communities of other countries. And so one of the problems of this is we're trying to build therapeutics or vaccines that really are based on the genetic material of one group of people. And there's no guarantee that that design will be fit for purpose, for example, our Indigenous community. And so now the scientific community is much more interested in this diversity, the differences between people, and the whole genetic code and how they might be different. And to be able to use that information to actually mine and design and model how we might cater to other communities, in particular for us, a streptococcus vaccine to deal with rheumatic fever or rheumatic heart disease that occurs in our Indigenous community and in the New Zealand Indigenous community. And their major histocompatibility complex, which is a genetically encoded determinant of protection, is different to, for example, mine. So it's really important that we're including the right components in our vaccine so that we can make sure that we cater to the most vulnerable group that we have in our community. So this is a step up from personalised medicine because it deals with groups of people that we want to protect for various reasons, but it also allows us to do some personalised medicine, get closer to that situation of individuals or individual families.
Yeah, just wanted to complement and emphasize that the benefit is obviously for these minority populations, but it's actually a benefit for everybody. They're nowadays, this sort of pharmaceutical companies, I was trying not to make sure I mention them by name, pharmaceutical company in the US who's investing a lot of money into sequencing populations, indigenous populations around the world, and the bet there is that there will be, for example, identifying genetic variants with strong effect, which can be potential drug targets. But because those variants in those populations are more common, well, we can identify them, and that at the end of the day will benefit everybody, so not just those minority populations.
Kirsty, what about the personalised medicine side of things? What's the promise there?
Yeah, I mean, I think where I work in the field of infectious disease, and I always think that the idea of personalised medicine and infectious disease has always been a bit behind the times, because people think instinctively, okay, you have a virus, you get an antiviral, and if I give you an antiviral, that'll work the same as if I give you an antiviral. And unfortunately, it's not that simple. It would be nice if it was. And so now what we are seeing in the context of infectious disease is that really we need to incorporate large amounts of data about different aspects of an individual, some of which is the genetic information, some of which is their clinical history. And I'll give you an example of this. So we're doing work at the moment in long COVID. And you've probably heard a lot about long COVID. It's a miserable condition. And probably one of the worst things about long COVID is there's no diagnostic test. So the diagnosis for long COVID is you had COVID, you have ongoing symptoms, we don't know what causes them, it's probably long COVID. And that's a disaster for the patient population, because a lot report being sort of gaslit by the medical community, saying it's all psychological. So what we're doing is actually, across a large number of individuals with long COVID, doing this really comprehensive characterization. So it's genetic characterization, it's immunological characterization, it's characterization of the virus that they were infected with, it's characterization of their clinical history. And what we can do now in the wonderful age of machine learning that goes beyond just ChatGPT is we can actually say, okay, let's put in thousands and thousands and thousands of features and see which of those features actually has a predictive value, such that we can develop an objective diagnostic test for long COVID. So it's really, I think, about integrating our genetic information with all the other information that we can obtain now with the technology that's available to actually do more comprehensive and personalised diagnostics.
Well, Trent, you've worked with some of these giant data sets. The promise and the reality were a bit different. How's it evolving?
I'll give you an example. So one of the drugs that I worked on, the original mutation that was discovered, or the genetic change, I should say, that was discovered, was a aerobics teacher in Texas who had incredibly low cholesterol. And there's one of many case studies, but that's the one that's often cited. And so they were able to actually work out, well, what was the reason? This person had super low cholesterol. And then they were able...
Wasn't the aerobics?
It wasn't just the... It turns out it wasn't just the aerobics and the salads and everything else. But, you know, and probably being in Texas, that wasn't the diet at the time. But... And look, so you know what the pharma industry was able to do is go after that particular change and figure out a whole new pathway about how cholesterol is controlled. And now there's a range of new products on the market that are available to people, especially with those people with something called hyperfamilial cholesterolemia, where they're really unable to control cholesterol levels with typical drugs like statins. There are many, many other examples. And then at the same time, the company I was working for had procured the entire genome of every individual in Iceland. Sounds a bit strange that you can buy all of the genetic material of Iceland, but that's what happened. And we decided that we would try and find a better drug. We would go after a genetic change that was even stronger, even more powerful. And we spent a bunch of time and a bunch of effort and a lot of money going after that target. And we couldn't mechanistically work it out, but we decided we'd test it in humans, and it didn't work, is the story. And the reason was, when we looked at a broader data set, and in fact looked at large genomic data sets outside of Iceland, that correlation no longer held up. And so I think it's just a really important, you know, maybe obvious learning of something that we've sort of touched on, is that while we've come a long, long way and we can gather a huge amount of this information, how we actually process it and use it is really important. And we're just at the start of that journey, per the research that's happening.
Are you saying that Iceland is just too small of a country?
It was more that I think there was so much excitement about the strength of the signals and the size of the data set that some of the other co-founders weren't properly taken into consideration. And I'm oversimplifying a little bit, but you know, those kind of learnings we need to be really, really careful of. One other quick example: there's a company in the US called 23andMe that many people will have heard of. You know, today, or I think it was overnight, they announced that they're stopping all therapeutic development. And so they have one of the largest human genetic databases on the planet, albeit a sort of different type of genetic data. And they were trying to use that to bring drugs forward. And they've just canned that entire part of their portfolio in their company simply because it's tough. And so, I
not
think...
for ethical reasons.
Not for ethical reasons.
That was a concern.
And they went through a lot of challenges. And I think, obviously, ethics of genetic information is a really hot topic and something, that we have to pay very, very close attention to. But this was simply an inability to use the data effectively.
Loic, what kind of strength, like how big would a database have to be to have the sort of predictive strength that Trent thought that they had when they were looking at Iceland?
Big. Really. Yeah. It's hard to answer the question. I think so. We have a little bit of a precedent. So it was not studying a disease, we were studying human height. And in my line of research, we often think about height as a model trait with this sort of very simplistic idea that if we can't even figure out how height is working, well, good luck figuring out what is happening for other complex traits or diseases. And for a trait like height, we needed approximately 5 million people including about 20% of them from all over the world, not just white Europeans. And so I guess, we're contemplating data sets of 20 million people. I think this is what we need.
Gabrielle, you were saying before about having these conversations about expanding data sets, expanding the genetic diversity of data sets, really wasn't something that we were seeing until COVID.
Yeah, that's right. I mean, it was actually quite difficult to publish some of these data because people were not really interested or the populations were quite small. I think now we're really intrigued for a whole variety of reasons to understand the genetics of different populations. As Loic said, learning from even small populations can really assist us in understanding how disease processes or resistance occur, just the same as if we have a single mutation that causes diseases in an individual child or a very, just 100 or so children across the world. What we learn from some of these really rare cases or small cohorts is really, really crucial to what we understand about the bigger cohorts. And so I think scale is important, but we shouldn't dismiss the small populations that are really unique resources and genetic libraries to assist us in understanding how diseases, resistance to different sorts of infectious organisms and comorbidities occur in predisposing us to different types of, for example, pathogen infections.
It's really exciting to hear how much has been done and how far we've come. I'd love to sort of look to the future. Trent, so much of your work is about looking forward, predicting, responding to threats as they arrive. What do you sort of see coming down the pike in this space in the next 10 years?
Well, I mean, one of the things that we probably haven't been as exposed to here in Australia, but is incredibly prevalent and on the rise, say in the US, is the use of complex genetic testing to aid both diagnostic for a health practitioner, but also for an individual. So if you're in the US, you can order yourself, I think it's about $2,000, a test called the Gallery test, which will basically look at a whole bunch of markers and your predisposition to cancer. And that's been quite successful. The other thing that they're doing is that they've now got a genetic test which is able to predict your risk of colorectal cancer much more accurately than saying what we do now, which is essentially look for signs of blood in stool and some other markers. And so these things are happening. And so as much as we're saying that there's challenges in a range of indications per the sorts of cohort studies that Gabrielle was talking about, we have the power now to very accurately predict people's risk with a high probability and let people take that proactive approach to their healthcare. And so what we know about the healthcare system is that, the journey that we're on in terms of the cost is unsustainable. Like it's clear, anyone can look at that, or you can go to any emergency room in the country and see that. our ability to move towards prevention-based healthcare and understanding how and when we need to seek medical help is probably really the future. That's one piece. And then just the other piece, just to finish, is that the tools we have in our toolbox have expanded dramatically. So, you know, the use of endogenous medicine, the use of cell therapy, the use of more advanced therapeutics.
What's endogenous medicine?
So what that means is, you know, typically what a drug has existed in the past is a pill, something like, you know, let's take paracetamol, where it's a small molecule that has a whole range of effects on your body. We use it if we have a headache or we have some kind of a pain, and it kind of works for some people. Whereas in the cancer field now, we can actually take cells from your body, reprogram them to direct them against the cancer that you have, and then give them back to you to go and attack that cancer. And in fact, you know, there's evidence in some patients that that may even approach a cure for those individuals. So that endogenous medicine approach is quite remarkable. We're now able to also do endogenous gene editing, where we can actually take things that we make in the lab and actually change your DNA within your body to correct for things like sickle cell anemia and other genetic diseases that are relatively simple traits. So this is happening. It's happening now.
I think one of the other really big changes is the fact that we now have the capacity to deal with big data, because, you know, we're talking about the need for diversity and inclusion within the data and representative data sets. But it's only relatively recently that we have had the analysis techniques, and a lot of that comes from machine learning and AI, but also the actual software and hardware capabilities to do this. And I think one of the really exciting spaces to watch this in the future is to see how quantum computing will sort of revolutionize all this analysis, because already we're seeing AI is changing how we interpret data, and quantum computing will be the next big jump.
Gabrielle, did you want to add something there?
Yeah, I think COVID really taught us what we can achieve if we use our imagination and really reach for the stars and collaborate, where we have those barriers that often interfere with us getting and exchanging samples and getting sequencing done and pass them all around the world. And so it's given scientists really big dreams to really take these technologies and be able to implement them across a range of different diseases. COVID for the first time allowed us to sequence viruses as they were evolving, and then to think about vaccines and use modelling and genetic sequencing to really identify what potential good targets might be, so that everything's not trial and error, which is a very slow process, and is what we did well before COVID. Now we can use these sort of high-end modelling, AI technologies, and the genetic sequence to start to pinpoint really what are the key targets we should be driving at, and how do we get to them faster. And we saw with COVID that we did get there faster, but the virus is evolving while we're getting there. In real time, it's challenged us with how do we keep pace with that, and how do we develop the technologies and the capability around the development of that data processing so that we can be ahead of the curve. I think it's really exciting.
Just the volume of data around, I think it's hard to understand if you're not really tapped into that space and looking for it, just how different it is to even 1 decade ago, Kirsty.
Yeah, absolutely. I mean, if you think back to the previous pandemic, which no one remembers, but it was the thing, 2009 swine flu pandemic. For those of us who had the virus, it was definitely a thing. And, you know, probably throughout that pandemic, we sequenced maybe 2,000 viral isolates. I don't know how many COVID isolates we've done, but I would say millions at least. I mean, I know probably within UQ itself would have already done thousands. Like, it's just night and day in terms of the data that's available, and that has real implications for, as Gabrielle says, sort of updating vaccine design, but also understanding the process of evolution. There was a lot of assumptions that were made about viral evolution in the early stages of the pandemic that were made based on very old, archaic theories. And it turns out when you actually monitor the evolution of the virus in real time, it's something different.
Recently, I was discussing with a friend of mine, and I'm trying to give him a sense of the size of the data we were dealing with. And I guess the best analogy, so this is me trying, so it may be a complete failure, so indulge me. But the best analogy I came up with was if you sit in front of, you watch a movie, about 1 hour movie, and you watch 4 hours of that movie every day for 5 years, then you will go all the way through at the end of that data set. So this is the kind of data we're talking about. Right? Imagine someone's time dedicated to watch a movie for 5 years, 4 hours per day.
What movie would you choose?
Gattaca.
Okay, so coming to that, this huge data set, incredible power, it's so exciting, we can use it for good. Could we use it for evil? What are the ethics around this? What are the safeguards that need to be in place so that the promise of all of this is realised and that it's realised with equity? Gabrielle? Fix this for us, please.
Yeah, simple.
I think the ethics around using genetic material, even in the pathogen space, is really important, and researchers have been very cognizant of that for many years. For example, engineering viruses that actually could do harm. And in Australia, some of those viruses have been generated and they never saw the light of day because it was quickly realised that they would be very harmful if they ever got into the community. So it is up to us to have those discussions and to really debate them out and understand what the boundaries are for the use of this genetic material. Other people can speak more widely to, for example, in the clinic, how we use genetic material and how we use that responsibly. But as scientists, it's really important for us to have that front and centre in how we think about the material that we get as samples from people to do our analysis, but also how we have that for the future and don't discriminate against people based on what we find in those analyses.
I think one of the things, so we've sort of been discussing this about the use of genetic information in diagnostics and how you can incorporate that with AI and make sure you do it in an ethical way. And I think one of the things is that, it's really about setting up large databases of consenting individuals who all know that their genetic information is there, it's de-identified, and that can be used to develop models, to develop whatever you need, such that when an individual patient comes along, you can plug in their data, the model will say, okay, based on this, you have a 70% chance of this diagnosis, but it's not that that data is being added to that data set. So it's about separating the data that we're using essentially for training versus the data that's being deployed for clinical information.
Flo?
Well, yeah, I totally agree, but I was just thinking this is quite challenging. I guess we had issues where you have identical twins, and one decided to enter a study and consented, but they are consenting to sharing the other twin DNA, right? And so I guess it's more of a comment. I don't have the solution, but this is a challenge, right, when it comes to genetic data.
Yeah, absolutely.
I do want to go to as many of your questions as possible first, but just before we do that, so start thinking of them. I'd love to hear from each of you what you think the biggest payoff might look like in this space in the next few years. Gabrielle's got a very thoughtful expression, so I'm going to start at this end. Loic. Where's the biggest payoff going to come?
Oh, it's hard. I guess for me, I'm excited. Trent talked about prevention. I think this is where we have the possibility of making a difference. So one number. So there is about 25% of preventable heart attack today that are associated with people with no prior risk factors. And we believe that with genetic data, essentially by skewing people based on their genetic profile to do sort of early screening, we can potentially capture half of those preventable heart attacks. So yeah, I'm really excited about this. So their current studies are trialing and measuring what I'm talking about, but this is going to be a major.
Kirsty?
I mean, yeah, I completely agree with the preventative perspective. I think from my perspective, it's seeing genetic data integrated into diagnostics for more accurate diagnostics. So, for example, one study that we did during the COVID pandemic, is we could actually look at individuals with COVID and look at what genes were being switched on in their blood and compare them to healthy individuals, and you could see obviously that there was a lot of genes associated with inflammation and an antiviral response. But what we could actually do is whittle that list down to 7 genes and use the expression of those 7 genes to actually predict whether those patients in 10 or 15 days' time would go on to develop a secondary bacterial infection. And that's really, really key, because when we talk about antibiotic use in viral infections, we want to be really careful. You don't want to give everyone with a virus antibiotics because they don't work against viruses. So you want to have that sort of antimicrobial stewardship. So I think it's using all these advances that we now have in terms of the technology, the availability of the technology, the cost of the technology, to really integrate that into standard clinical diagnostics.
Trent?
Yeah, I agree. I mean, I think it's matching the person to the treatment, or matching the person to the journey that's going to maximise their outcome to better health. And so I think that's the really exciting piece of this. It's a very imperfect world to get a diagnosis and then be forced to take a drug. I don't think that's the way that we should be thinking about the treatment of disease more holistically. And so our ability now to use these data sets to benefit people, to extend lives, to have people living more healthier lives, that's what I'm really excited about.
Do you want me to pause for a sec so you can quickly call copyright and just matching the person to the journey that's going to link them to better health? Someone in this room is going to copyright that before the end of the night.
Can someone get that website?
Gabrielle?
Yeah, so I think there's 2 things that I think we'll have some massive wins for. The first one is preventative healthcare. And a few years ago, we had a discovery in the lab, and I said, well, if we could get an insight into how these cells work earlier, we could potentially prevent, for example, melanoma. Mm-hmm. And so that was a blue sky thought at the time, but concurrently, technology to be able to see changes, small changes in the skin, have been developed. So now this journey really is a reality. I think the second thing is that we have a lot to discover about what cells are doing, how they're responding, how the viruses and the bacteria are interfacing with the bodies. So the ability to understand the genetic code of those organisms as well as the cells will really uncover pathways that we haven't begun to understand. Many of the treatments we have at the moment, for example, immunotherapy, it's transformational if it works in you. But we have major gaps in our understanding. So really understanding what those other pieces of the puzzle are, so that now instead of delivering, say, a 30% curative rate to people, we can increase that to 70 or 90 or 100%, is sort of the holy grail. And the exciting thing is that these are new targets that we're not considering now, which will make new pathways to actually develop different types of therapeutics, which hopefully will lessen that trajectory of the public health cost.
Amazing, thank you so much. Now it's your turn to ask some questions. I've been hogging the mic. Marla's going to run around with this mic, and I'm going to jump on Loic's one from before.
Tegan, I actually do have a question that was submitted earlier on. How much does methylation of DNA play into the disease question, and is there any method to undo the effect?
I mean, this relates to what we were talking about earlier on, and this was the idea that environmental stimuli can actually determine which genes get switched on, which genes get switched off, and how that's passed to the next generation. So the mechanism by which that happens is actually methylation, at least as one of the mechanisms. And there are certainly drugs that people are investigating to see if you can actually change that methylation profile and then change what genes are being switched on and switched off. So as a shameless plug for my own research, this is actually something we're looking into for long COVID, where we can see that a lot of these individuals with long COVID have a lot of these pro-inflammatory genes switched on, and it's causing this chronic inflammation. So now we're looking to see if we can get drugs that switch off those genes that can actually alleviate some of those patient symptoms.
Could I ask two questions? Has plague left a genetic footprint in the population of North Europe, which might indicate susceptibility to resistance? And the second is, are there any subgroups in Australia in which the recommended paediatric vaccine schedules don't work?
Oh, can I take this?
It's like set up for you, I think, that one. Yeah.
This is like my question. So I'm super into ancestral pandemics and what has changed in the human population as a result of these pandemics. So there's been a few theories around plague, nothing that has been comprehensively shown, but I will give you an example of something that has. So in the Caucasian population, the risk of being a carrier for cystic fibrosis is ridiculously high, 1 in 20. It's very, very high. And the theory as to why that's happened is actually linked back to the cholera outbreaks that happened in Europe. And the channel that is affected in cystic fibrosis, that means people have a lot of mucus in their lungs, that actually helps control salt regulation in the gut. So it's thought that individuals that were heterozygous, so they had one copy of this defective gene, actually had an evolutionary benefit in the cholera outbreaks because they could survive that dehydration and diarrhea and that process. So absolutely, past pandemics and past disease outbreaks have shaped our evolution, and there has been a lot of discussion as to what we will be like in 100 years and how COVID will have impacted our own evolution, because that was, again, another massive event. Can you remind me what your second question was, because I got too excited.
Pediatric schedules.
Sorry?
The question wasn't cholera, the question was plague.
Yeah, no, I know that. I just didn't have a great plague example, so I tangented into cholera.
The second one was, are there any subgroups that don't respond to the recommended paediatric vaccines?
So generally when we've studied, in general, paediatric immune responses are much poorly studied compared to adult responses. We do know that a lot of the comorbidities that we see in adults that affect immune responses also affect kids. So one of the key examples there is obesity. We know that obesity can affect your immune response to vaccination as both an adult and a kid. Whether there are children, specific children that are absolute non-responders to a specific vaccine, I mean, probably statistically yes, but there's nothing that's really mandated in terms of the vaccination schedule to address that. So we do know that there are factors that influence the paediatric response.
Wonderful. Listening to you.
Oh, thanks. It's great insight into some genetics that I'm learning now. But as far as the epigenetics, which almost seems to be fashionable enthusiasm at the moment, I was just interested in finding out how local epigenetic changes get transferred to germ cells into the next generation. Is that sort of speculation, or is there actually hard data on how that all works?
Well, I mean, the standard theory is that, you know, the epigenetic marks should be wiped, right? And that happens during germ cell formation. And so I think, you know, what's not clear is what are the other signals within those cells that are persisting, that are allowing those marks to come through to the next generation. It's absolutely clear that they do. And, you know, I think that the science is early on why. I don't know, Gabrielle, if you've got a better answer, but I mean, it's, you know, there's plenty of studies that are starting to look at, you know, the persistence of those marks. Some of the technologies we were using when we looked at that weren't as good as they are today. And we're seeing higher resolution ability to look at some of those marks.
Yeah, I don't perhaps have a better answer, but even not between generations, when we look at some of the cells, the immune cells, they turn over very quickly. And so there's this continuous question of how you maintain that trained immunity or immunological memory and the epigenetic imprints of those cells to maintain the protective barrier. And as yet, we don't know what the mechanisms for that are, except to say that they're very important in that protective mechanism that we have that keeps us healthy every day.
So those sort of studies looking at the changes transferred, are they in large animals, bacteria? Are they?
Oh, go for it.
Yeah, I mean, there are also studies in humans as well, where you're looking at generational expression of various epigenetic markers, especially after major events like a famine or things like that. So there are studies that can be done in humans, and it just takes multiple generations to see how things are conserved.
Yeah.
Yeah, if I may, just to complement, I think to make the picture even clearer, well, probably not, but it's going to make it more complex. But some of those epigenetic marks are themselves controlled by our sort of hard-coded genetic information.
Yeah.
So which means that when we look at maintenance of those epigenetic signatures across generations, part of that maintenance is actually directly coded in our DNA. So if I have a particular genetic variant that is controlling how much that particular gene is methylated, I'm more likely to pass it to my children, etc. So I think Kirsty was talking about how the environment is changing those epigenetic marks, but our own DNA is also contributing to that.
Yeah. So there's still lots of questions in between, isn't there?
Yeah, absolutely.
Yep. Thank you.
Hi. Since the Second World War, there's been a decline in the burden of infectious disease and a commensurate rise in the burden of autoimmune conditions, and often there's no treatment for these. What sort of hope is there for being able to improve treatment in autoimmune conditions?
I mean, I can start this from the infectious perspective. What we're appreciating now is that actually those things are not separate conditions, but often inextricably linked. So we're realising now a lot of autoimmune conditions can be triggered by viral infections, for example. So we now know that viruses like EBV, Epstein-Barr virus, which causes glandular fever, is absolutely necessary for the development of MS. You won't have MS if you've never had EBV. Similarly, we know EBV plays a role in things like lupus as well as other various autoimmune conditions. We see a rise in autoimmune conditions like type 1 diabetes, very clearly correlated with COVID. So again, I'm biased because I work in infectious disease, but I think that actually by targeting some of these infectious diseases that we haven't gotten under control yet, things like Epstein-Barr virus that's just rampant in the community, that will actually lead to decreases in autoimmunity.
And the other really interesting component where the science hasn't got the problem solved, and Gabrielle touched on it before, is around the other parts of our body that control our metabolism, immune system, mental state, and that is the bacteria and viruses and fungi that live with us, our microbiome. And that interplay for how the microbiome trains the immune system is likely to have a really important role in the emergence of many autoimmune disorders. And so we know through modern living, through more cleaner societies, whether it's antibiotic use, there are many reasons that our microbiomes have changed and we've lost a lot of diversity. And so, that could be an additional factor that's contributing to some parts of this. I mean, it's definitely not a slam dunk, but people are starting to understand those relationships as well.
Yeah, thanks everyone. It was really engaging and motivating. My question's an open question. It's actually, I'm going to keep it one word, just so whoever wants it can go for it. Of course, in the context of the topic, didn't hear much discussion, wondering it's the elephant in the room in my mind. And my one word is insurance.
Yeah.
I want to take this. Insurance. I think Australia is doing quite well in that space. I just want to say, I don't know if you've seen that in the news, but probably about 2 months ago, the government announced its ban for insurance company using a sort of negative result from genetic testing to discriminate consumers. And so I think we'll need this to be replicated across the world, because that has been a major obstacle for people even engaging with this type of research. But yeah, I think it is a big elephant in the room, but that elephant can be caged.
Thank you, Tegan, for your expertise and also for helping our panellists communicate complex science and share their expertise in a really accessible way with all of the people in the room tonight. And really thank you for giving your time. And as you've seen tonight, there is powerful research on the way and a lot of hope and interest in the future of medicine, particularly infectious diseases, and ways that we can tackle diseases moving forwards in the future. And we've come a very long way, and I think the future is pretty bright. And thanks to all of you for the research that you do and the interest you take in improving human health for the future.